Hello all, and welcome to the first Patient-Centric Sampling Interest Group webinar of 2024. My name is Neil Spooner. I'm the chair of the Patient-Centric Sampling Interest Group, and today I'm really excited to bring you Mitch Johnson, the lab director of Velocity Labs, to talk to you about analytical considerations, tips and tricks for quantitative analysis of patient-centric samples and microsamples. Before we start, I just want to thank our partners and sponsors who provide the funding that enables us to put on these webinars and the other activities of the Patient-Centric Sample Interest Group. You can see from here who our sponsors are, and just we'll have a Q&A session after Mitch's presentation. If you'd like to use the chat function to submit your questions, please do so. And please note that the webinar is being recorded and will be available on demand for free afterwards through the Patient-Centric Sampling Interest Group website. So with that, I'd like to hand over to you, Mitch. I just need to stop sharing and go off stage and hand over to you and see
Excellent.
you outside.
Thank you, Neil. Let me just share my screen really quickly. Excellent. Well, welcome everyone, and thank you, Neil, for the introduction. It's an honor and a privilege to be here. And those of you that don't know, Neil Spooner is considered one of the godfathers of quantitative bioanalysis for microsampling. So it's a pleasure to be here, and thank you for having me. My talk today is about analytical considerations, tips, and tricks for quantitative analysis of patient-centric samples and microsamples. A little background about myself. I earned a bachelor's degree in chemistry from Monmouth College in Monmouth, Illinois. Then I pursued a PhD in analytical chemistry from the University of Missouri, Columbia, under Dr. Mike Greenleaf, where I focused on LC-MS analysis of natural products and botanicals. I started my industry career at Alliance Pharma in 2017, which is now known as Resolian, working on a lot of regulated bioanalytical work. That's where I really broke into the quantitative bioanalytical space. And then I took an opportunity and joined what was then known as Seventh Wave Lab, now as Innovative, and was there for 3 years doing GLP bioanalysis, DMPK ADME. I had a short stint at a small biotech company called Biomed Valley Discoveries, where I was a project manager outsourcing BioA, and then got together with some individuals, some like-minded individuals, and co-founded Velocity Labs in 2021. And currently I serve as the lab director here. So overview of the talk today. First, a lot of you are probably familiar with patient-centric sampling and microsampling, but I'll give a little bit of a background on that for those of you that may not be. Then I'll talk about method development workflows that are specific for these types of samples, and then additional considerations that we have when we're validating methods to be compliant to support GXP bioanalysis. Then I'll go through a case study where we implemented patient-centric sampling, and then talk about some future outlooks of this topic as well. So the question is, we get a lot from our collaborators, is why would you use patient-centric sampling and microsampling? So if we think about just conventional blood sampling for a clinical study or even just an animal study, this is going to first and foremost require you to have a phlebotomist or some type of highly trained in vivo person to take the samples. And you're also going to be collecting a relatively large volume of blood. This is also uncomfortable and painful for a lot of people. I know I've given blood and I feel queasy afterwards. It's just not an optimal way to collect blood. And also, when you're withdrawing that much blood, you're disrupting the body and things like that. It's also very costly and time consuming for the programs. When we think about patient-centric sampling or microsampling, we're just at its core, we're following the 3 R's. We're replacing, reducing, and refining. This is also minimally invasive. So both from a pain and comfort standpoint, we're not disturbing either the human or the animal as much as we are with the conventional blood draw. It allows you to replace sparse sampling models with, let's say you have an expensive xenograft model that you're using or a tumor model that you're using where the animals are very expensive. Now, instead of having to have an N of 18, you can have an N of 3. It can also improve your clinical trial enrollment. So decentralized clinical trials continue to become more common. And so having a patient that can collect a sample at home allows them to maybe enroll in a trial where they would normally not be able to drive to, or for a patient that might be very sick and just is bedridden, those kind of things. It also affords you to potentially get a richer PK and TK data set by being able to collect more samples more frequently, you can get a better picture of your ADME properties of your drug. And then another application is pediatric diseases where there's not a lot of blood to be taken, or you're just doing intravenous collections isn't as feasible as it is with adults. So just being able to collect a few microliters of blood from a pediatric patient. So what has made this possible? So in the last 20 years, there's been some advances both on the sampling side as well as on the analytics side. So this whole thing really started with dry blood spot analysis, which predated me in bioanalysis. But that just involved kind of, and I'll touch on this a little bit more, but involved using a lancet and dropping blood onto a piece of paper and then sampling it that way. And then that was advanced into what's called VAMS, which I'll talk on, volumetric absorptive microsampling, some additional microfluidic technology that allowed better microsampling devices to be on the market. And then there were significant advances also in the analytics as well for detection and analysis. So the picture on the left is what a dry blood spot card typically looks like. And so a patient would take a lancet and do a finger prick and then apply multiple blood spots on this card. And there were some issues with this across a couple different areas. First and foremost, with the samples sometimes weren't homogeneous. So the bioanalytical scientist would take a punch and punch a portion of that spot out, and you could have actually different amounts of analyte concentration throughout the blood. Is also plagued by hematocrit bias, which the different hematocrit in the blood could affect the concentration measurements. And then you have things like contamination, where a blood spot kind of going from the one space to the next space. So although it was kind of the breakthrough to allow some patient-centric sampling and microsampling, there was room for improvement. And that's what really brought in VAMS, which is to the right. So it's the same concept. We're still doing the dry blood spot, but now we have this cartridge that has technology built in that will essentially collect that sample in a volumetric fashion. So like on each one of these tips here, that tip is collecting 10 microliters of blood with precision that's comparable to a volumetric pipette. So that was a big improvement in the technology and collection. And then also on the analytics side, I'm showing some pictures here of gold standard triple quad mass specs that are used in the pharmaceutical industry, ABSIX. In the year 2001, the API 4000 debuted. Great instrument. It's a workhorse. And then now we have access to the 6500 plus, which has been significantly improved in the last 20 years. So as you can see, the two biggest differences are the 4000 utilizes the linear quadrupoles and has a different source than the 6500s. Back when the 4500 was debuted, they re-engineered the geometry of the quadrupoles, which helped filter out some additional background noise. And also there were significant improvements to the ionization source to increase ionization efficiency and ultimately produce more ions, giving us more sensitivity. This is just to show you a comparison. This is the same molecule on the two different systems. At Velocity, we use both of these systems. They both are, have their purpose for different applications. On the left is a chromatogram of a 1 nanogram per mil extracted sample on a 4000, optimized for that instrument. And on the right is a 1 nanogram per mil chromatogram optimized for the 6500. And you can see easily about a 20-fold signal-to-noise improvement. This is going to vary from analyte to analyte, but we routinely see anywhere from 10 to 50-fold improvements when comparing the two. So from a collection, typical plasma or blood collection of maybe 100 microliters that would need to be used on a 4000 to achieve that sensitivity, that can be scaled back to maybe 10 microliters and allow microsampling to be implemented in the place of traditional plasma collection. These are a few of sample collection devices that are available just to illustrate. So on the left, there's a Capitainer, and this is quantitative dry blood spot. So this is really the second generation of dry blood spotting. So it took those cards and then implemented some of that technology to make this more, it's collecting a quantitative volume, and then that entire portion of that card is analyzed. The middle is a microsample. So the very bottom of that tip is the VAMS technology, and that would be what would be applied to either a droplet of blood on the finger or a tail nick from a rat that would be placed on the sample and collect as well. And then the right is the Tasso M20, which is optimal for clinical studies. This device is placed on the arm and pressed in. It makes a minimally invasive puncture and basically allows blood to then collect into that cartridge as well. So after the background, we're going to talk about specific method development workflows that are very critical to microsampling. So the first part of the development workflow doesn't really deviate from traditional plasma. You would, get your analyte in solution. You would first tune the mass spec for optimal parameters for detection. Then you would develop reproducible chromatography, so you have a sound LC-MS method. Then what we like to do is first test our extraction in wet whole blood just to make sure we don't see any interferences from that matrix, as that's going to be the same matrix that we're going to be using for the microsampling. Just making sure that we have our chromatography and our method is still reproducible and linear. And then here's a big one that's often overlooked, is doing stability assessments in the wet whole blood. There's going to be a period of time that blood takes to dry from wet to dry after it's collected and processed. So you're going to want to ensure that analyte does not degrade during that process, or that will throw off the results. And then the biggest one, in my opinion, is extraction efficiency. There's several variables here to make sure we're actually extracting the drug off of these devices, as opposed to maybe just, crashing them out with an organic solvent. We're pulling these off. With that comes a lot of variables. So that's very paramount. So what we've developed and what I recommend is to kind of have a go-to extraction and recovery system that tests a couple of different solvents, modifiers, things like that. And it's an iterative process. So the first thing is selecting the appropriate solvent, which is going to be dependent on the log P of your molecule and the pKa of your molecule and things like that. We found that methanol across several different scaffolds works very well. Acetonitrile also works well. And then as we go down the list, we see harsher solvents like MTBE or isopropanol. That's going to, as you get stronger, you're going to pull the analyte off, but you're also going to pull other constituents like the blood off of that device as well. So methanol seems to be that nice fine line where it's strong enough that it will extract the analyte, but will not cause a lot of the other blood particulates to come as well, which can cause matrix effect, interferences, things like that. The pH can also have a big effect on the recovery. So we'll usually take those solvent systems and look at them at neutral, basic, and acidic pH. As you go harsher in either direction with the pH, same thing as the solvents, you get additional components that come off. So if neutral works best or is equivalent, we tend to go with that, as it will be more selective for just pulling the analyte off. You can also have some assistance when you're doing this. We found that sonication works well. It just helps kind of disrupt that and really remove that analyte. But sometimes just vortexing or shaking will be sufficient. And then time is also critical. So we'll test a time period, usually over the course of an hour, to make sure that we're going to completion. We're not doing it for too long where we might see some degradation. And there are some other strategies. You could also incorporate heat or pulsing. There are some devices that will actually send electrosonic pulse through that might help with that extraction. And then pre-wetting is another technique. Those blood samples are going to be dry. Some of them might even be crusty. So if you have a step where you just add a small amount of water to help pre-wet and then add your extraction solvent, if you're having difficulties, that's also another strategy too. So here's kind of a plot of two experiments that we usually perform. One of them is just drying time. So as I mentioned before, you're going to add these samples and the blood is going to be wet. And then over the course of maybe a couple hours, it's going to dry. And of course, we want our standards and QCs to match our patient samples as closely as possible. So the patients are going to be collecting samples, putting them into a package, usually with desiccant, and then sending them to the lab or central lab. So those samples are going to have, before they're analyzed, they're going to have plenty of time to dry. And as you can see, these plots are just analyte response peak area ratios as a function of time. We see better extraction efficiency while the blood is still a little wet. And as it goes down, it stabilizes. So our traditional approach is just to let the samples dry overnight as we're doing initial method development on some other conditions. And then we'll test this as well over a longer period of time to make sure that as those samples dry, we don't see a loss in recovery. And the same thing for the sonication time. The figure on the right, you'll see as we reach about the 10-minute mark for sonication, it about plateaus. So this method, I believe we used a 30-minute sonication time for it, but then it further doesn't significantly degrade afterwards. So finding that spot where you're going to have reproducible and reliable recovery based on these two attributes is very critical. So now I have a few videos that I'm going to show just demonstrating kind of how we go about preparing the samples that we're going to use to support the sample analysis.
When preparing standards and QCs for microsampling, your
you're first going to take your analyte in solution and first spike it into the matrix, which in this case we're using human whole blood. So we will first fortify our HQC sample by spiking directly into control human blood. And then we will mix that. And typically when we're doing this, we'll allow some time for it to distribute and equilibrate into the whole blood. But for the sake of this demonstration, I'm going to go ahead and move on. After that's equilibrated, we would open the tube, and then, in this case, we're using the Mitra, we would submerge the tube into the blood just to break the surface and allow the capillary action to work for it to fill. Once it fills, give a 1 Mississippi count. Make sure to keep the tip nice and vertical, and you'll see that the tip has filled completely. Now I would take the tip. There's no leaking of any blood. I would put it in the clamshell to dry overnight. For the Tasso M20, it's a little bit more complex than the Mitra, but follows a similar workflow. So let's assume I've already fortified my whole blood with my analyte of interest. We're going to then load using a volumetric pipette. We found that 100 microliters is an ideal volume. And we're going to spike the orifice. There's two holes in this orifice. We're going to spike on the side opposite of the film. We're going to slowly insert the pipette and allow that microfluidic technology to allow that blood to flow down on the different cartridges. And you'll see once it gets past that fourth cartridge, there's a little excessive way for it to catch the extra blood. As it flows through, once we see that blood at the bottom, we know that it's filled, and that's going to catch the extra blood. After that has come to a completion, we will remove the film on the other side of the device, which allows it to dry. Very simple to do. Then we will place the device into the rack, allowing it to dry overnight. Once the Tasso device has dried overnight, simply use this tool provided with the kit to open the device, which provides access to the cartridges. Using a pipette tip, then you can simply pop off a device into your extraction medium, then reseal the device. You have 3 replicates left for analysis. Excellent. So that's high level on what we go through as we're developing methods. And then in addition to that, with microsampling devices, there are some additional experiments and considerations you need to take into account that are different than traditional plasma specifically, but also whole blood. So the first one is hematocrit bias. Mentioned previously, the dry blood spot technique, the original one, was plagued by this. It can have a big influence. So we will still look at this because it can have an effect. So we'll look at a normal hematocrit for a healthy individual is about 45%. So we'll look at down to 25 and 65. That covers about all living conditions to make sure both on a loading front and an extraction front that doesn't influence the results that we're getting. Typically, these samples will be placed into a cartridge or an envelope under desiccant and sent overnight on a plane, in the mail, those kind of things, not on dry ice like typical plasma will be. So you need to assess elevated temperature stability. To cover this, we usually do 72 hours, and we put the samples in our incubator set at 40 degrees C, and then assess them as stability quality control samples. Something different is you're also going to do long-term stability at ambient temperature. These devices, although they can be stored in freezers, they're really, the optimal way is just at ambient temperature. So that's something that you typically wouldn't do with plasma or wet whole blood that you're going to do for Tasso. Another one is elevated or reduced humidity that just kind of goes along with that elevated temperature stability. There's not going to be as much control over the sample shipment here as there would be for plasma samples. And then just reproducibility amongst the device and between the devices. With typical plasma, we're pipetting. There's not a whole lot of variability there from vial to vial or from aliquot to aliquot. Here, there's more variables going on with just how it's loaded, how the patients are loading them, things like that. And then another one that we didn't really realize while we were working on this project until after we started getting patient samples, and this is also very important, is the effects of the device being overfilled or underfilled, which is demonstrated on the right, both with the Tasso and the Mitra. So the middle is the ideal fill. And this technology is very good. You kind of have to go above and beyond dramatically to get these effects. This isn't something that routinely happens, but there will be patients that do not comply and use the device appropriately, which is demonstrated on the left. They might overfill it, or on the right, somebody just may not have enough blood. They may not use it right. And so just kind of assessing, you may have a situation where an underfill means you have 3 devices that are perfectly filled and then one empty one. You could do an assessment to see if the top device still meets acceptance criteria. Obviously with the Mitra, probably not as possible because there's only one cartridge, and you can see that's just going to have less blood volume. So incorporating those into your experiments and then figuring out when you see those in the sample analysis phase, how to deal with those. Now on to a case study that we supported at Velocity Labs. So one of our collaborators came to us and wanted to implement the Tasso M20 into an existing clinical program for a small molecule. So their main objective was to look at the pharmacokinetics and of this device, which collects capillary blood, and compare it to traditional sampling where they already had PK going on. So the process that we took was we were going to develop and validate methods in wet plasma, wet whole blood, and then the Tasso M20. And in addition to that, we were going to determine in vitro whole blood to plasma ratio for a comparison. So now I'm going to show some results, just highlighting a few key attributes from the validation. So the top left here, we have plasma. We can see this is just one of the precision accuracy batches. Excellent low CVs, excellent accuracies from the validation. On the right is whole blood. Once again, very tight CVs, good accuracy as well. And on the bottom is Tasso M20. Still meets acceptance criteria. We noticed it wasn't quite as rugged as the whole blood, but I think that's to be expected. We're using the same LC methods. There are a few more variables that are going on. The method still performed very well, but I don't think your expectations of having it perform as well as plasma using volumetric pipettes is always going to be the case, but it's still sufficient enough to support GXP analysis. Just highlighting some of the additional tests that I mentioned, we looked at elevated temperature stability and then hematocrit bias. So the top table is just showing both with and without desiccation for 72 hours. We went maybe a little too far and tested things, but this was just to kind of cover, you're provided with a packet that has desiccation. Maybe a patient doesn't slip their sample appropriately in there and the sample is stored without desiccation. So that's just to cover some of these things. You're going to be dealing with real world cases, not professionals always collecting the sample. So it's good to just pressure test some of these things. And then on the bottom is the same thing. This was a combination of, does the hematocrit bias and the elevated temperature in synergy affect any of this? And you can see by the accuracy that the molecule was stable even under the hematocrit bias, even with the elevated temperature stability. We also established a little bit of a shorter long-term stability than you normally would, both under desiccation, without desiccation, and then in the freezer. This was just to kind of, once again, bracket and cover any sample shipping accidents that may have happened if something was left outside of a packet, or maybe sometimes the samples might get shipped on dry ice, just covering some of these different things that could occur in that shipment period. Once again, all of these things, none of these different conditions or stressors affected the accuracy of the quality control samples. And then the long-term stability. So this was very helpful to us. We did LTS, one LTS time point with hematocrit bias, and then once again, just under freezer and ambient. This allowed us to then store the samples for a longer period of time, and the LTS is ongoing here as well. Compound is very stable for 42 days, just at room temperature. And then also a thing to note is this is really the true testament of the method, is how does your method stand up during incurred sample analysis, not samples that are prepared under control in a laboratory, but patient samples. So you can see here using the same method, the plasma and the whole blood had excellent 95% pass rates. The Tasso still met acceptance criteria for the ICH M10, although there tends to be a few more what we call flyer samples where the percent difference from the first to maybe the second is outside of that 20% acceptance criteria. And I think this is a kind of a twofold thing going on here. We saw in the precision and accuracy, although it's still reliable and going to give precise and accurate, it's not going to be as tight as the plasma and the whole blood. And then in addition, there's more variability with the patient samples. You're not having one aliquot out of a tube. You're looking at multiple different samples. So small fluctuations from sample to sample or things like that could cause a little bit of a bias, but ultimately was still sufficient and quality enough to support and meet all of the GXP criteria. As I mentioned, we also wanted to look at the blood to plasma ratio. And why is this important? Well, we wanted it as a marker and a predictor for what we were going to see in the clinical study. So, a lot of people will use plasma to get their exposures, but plasma is really just a surrogate for, blood exposure, right? And the reason that people can do this effectively is a lot of small molecule drugs are really in that 0.7 to 1.3 blood to plasma ratio. So whether you sample plasma or you sample blood, you're going to get close to the same answer. That's not always the case. There are examples like sirolimus. It goes into to the blood. 95% of it will go into the blood, so you won't really see much in the plasma at all. So if you are going to sample plasma, this is something that's important to do no matter what. But even if you're going to do something like this where you move from plasma to blood, it's important to understand how those exposures are going to scale based on what compartment you're looking at. So based on the in vitro experiment that we did in human whole blood, we saw that the blood to plasma ratio on average over multiple concentration levels was about 73 or 74%. So that means if I have a plasma sample that's 1,000 nanograms per mil, that would equate to about 740 nanograms per mil if I was sampling whole blood. So the design of the clinical study. So this was purely designed to bridge the program from the existing PK in plasma and to see if the collaborator could then take this Tasso device into their future clinical program. So there were 10 healthy adult subjects enrolled. They all received just one single dose. And then over the course of 7 days, 15 samples from each compartment were going to be collected. So how this worked is the patient went into the clinic, they were given one dose, and then over the course of the time, an intravenous blood sample was collected. A portion of that blood was split, converted to plasma, and then immediately after that, a sample was collected using the capillary blood sample was collected on the arm using the Tasso M20 device. The patients then left after day 1, took a device home, did a self-sampling device on day 3, and then returned to the clinic on day 7 and did the blood, plasma, and Tasso collection as well. This was also just to work out any kinks with home sampling and things like that, and also to make sure that the profiles continued, what the profiles look like over the course of that time period. So here are the PK results from that study. So you'll see here plotted, this is analyte concentration versus time, and black is the plasma compartment, the red is the whole blood, and the orange is the Tasso M20. I've also added just the Tmax, the Cmax, and the exposures up here. First, let's look at plasma and whole blood. You'll see both the Cmax and the exposure for the plasma is slightly higher than whole blood. It came back about 80% or so, which aligned with our in vitro results. That's what we were expecting. But then also, and I think this was a bit of a surprise at first, the Tasso M20 exposures came back about on a Cmax level, about 30% higher, on an exposure level, maybe 10 to 15% higher. And we had a lot of conversations. Is there something going on? How do we explain this? And really, both of these answers are right. When you're looking at Tasso M20 or microsampling, a finger prick, things like that, you're looking at capillary blood. So this is a different compartment than intravenous blood based on how the drug is distributed to tissues in the body and how it moves through highly perfused organs and things like that. You can see differences in the exposures between these two compartments. But overall, you can see the PK profiles of the two drugs. There were no significantly huge differences between the different compartments other than subtle differences in the exposures between the compartments. So this was, in all efforts, a success. In the conclusion of that study, we successfully validated the methods for both plasma, whole blood, and Tasso, and the validation and the sample experiments all met acceptance criteria to be submitted to the FDA according to ICH M10. And I think the biggest thing was the collaborators successfully bridged that study, and they didn't have to redo any of their phase 1 data. They came up with the scaling factor between plasma and the Tasso M20. So if they knew they were getting 100 nanograms per mil in the Tasso M20 and they needed an IC50 of 50 nanograms per mil, even scaling that down to maybe 80 or 85 nanograms per mil equivalent plasma, we're still above that IC50 in the capillary blood collection. So we're still hitting our target. So that was a success that they could use. And because of these results, they continue to implement this into their phase 2 clinical program. So just some recommendations and some final thoughts here for if you're thinking about implementing this into your clinical program. I recommend implementing this early on in the preclinical phase. I don't think you necessarily need to do your GLP tox studies with it, but just make sure that, you might do a PK study with it, compare plasma to blood exposures. It's really easy to do. You can microsample first and do a satellite collection of plasma and just see, are these comparing? Are we able to get a good solid bioanalytical method with microsampling? These just confident is going to give you a lot of confidence to move this through and not have major surprises later on in the clinical. Also determine the whole blood to plasma ratio in vitro across multiple species. That way you can scale and bridge appropriately across the different study types that you decide to do. I would also highly recommend using a central lab. There's going to be some discrepancies, some samples that aren't filled appropriately, things like that. And these central labs are there's going to be spurious shipments and things. So having a central lab that then can collect all these samples in an intelligent way and then distribute them to the lab for analysis in batches of samples that make sense is going to make data reconciliation much easier and allow you to know of discrepancies early on. Maybe you need to replace a patient or a subject, things like that. Also work with the BioA lab that's experienced with this. There is some additional intricacies to this as opposed to plasma. There's additional equipment, there's additional experiments, considerations, things like that. And the labs that have experience and workflows with this will be able to support your needs much better. And also have a plan for some imperfect collections. As I showed earlier, you're going to have situations where samples may not be able to be included. How are you going to address missing data pieces in a PK data set? Are you going to need to replace patients? Are you going to be able to just do the profile without that time point? Have discussions early on about not really, if, it's a matter of when. And then also, be prepared if you take this later on, be prepared to come back and bridge the data as well. So my final thoughts on some technology and just future outlook on this industry. So the decentralized clinical trial space is continuing to grow. We're going to see this implemented. The technology is only going to improve. From our end, the sampling side has farly progressed quicker than what we typically have to support us on the laboratory side. So we would like to see some more robust solutions for just sample storage. Some of those demonstrations that I showed earlier, those racks that we hold the TASO devices in, we actually had those custom 3D printed to support those because there is not a really good commercial solution. So, storing samples and coming up with a good way to make that more traditional to the 96 well plates that we use for plasma would be super beneficial to the laboratories. And then there are some available. I know TASO has one with a plasma or a serum base, but sometimes whole blood isn't going to work as well. And it would be a shame to just limit a clinical study to that because you can't sample that compartment appropriately. So having some more plasma-based microsampling technologies would be ideal. And then the ability to incorporate some additives and stabilizers. With the current technology, we really don't have that ability. We do see increased stability with the TASO M20 and the Mitra and the Captainer and these things that are drawing the blood samples quickly. But there are situations where we might have a molecule or metabolite that's not stable. And in a traditional plasma setting, we could add a stabilizer to it to prevent that from breaking down. And unfortunately, we do not have that ability right now with a lot of the microsampling. And then if this is really going to take off, we're going to need to improve the throughput of sampling. So I showed some videos, and the processing can be a little tedious. And so to support large clinical trials, we're going to need some robotics or some liquid handling accessories where we can put in full clamshells of samples and maybe they can read a barcode and pop the tip off and process that. But that would really help this because there will be a bottleneck of supporting large clinical studies because it's a very manual and laborious process as it stands. And there are some regulations. There's mention of dried matrix sampling in the M10 guidance. It's very brief. It's not very descriptive. There are white papers about microsampling, but really having the BioA community in accordance with the regulators come together maybe issue a white paper or update a guidance document on and being more specific about these are the tests, these are the acceptance criteria, this is what needs to be done for the next generation of microsampling. So with that concludes my talk, and just some acknowledgments. I would like to thank PCSIG and the evidence base for inviting me to give this presentation, specifically Neil Spooner and Phil Garner for all their help in the background getting this up and going. My Veloxy Labs colleagues, specifically Dr. Shane Needham, who's an advocate of this type of work and really brought this idea to us a couple years ago, planted some seeds and allowed us to grow in this, and Colt Cookson as well, who's a colleague and was very intimately involved in the project and the case study that I showed. Our collaborators who implemented this strategy and worked with us as we were getting some of these things going, and the additional collaborators that continue to implement these as we gain more and more projects from this, and the vendors as well. Without this, everything from the improvements in the mass spec to the new technology for the microsampling devices and things like that would not be possible. So at that point, that concludes my talk, and thank you, and I'd be happy to answer questions at this time.
Thank you, Mitch. I'm just going to share my screen.
Excellent. Do I need to stop sharing then?
I think you already have done.
Okay.
I'm just trying to, there we go. So just if you can use, I see some people are already submitting questions through the chat function, which is great. Thank you. If you can continue to do that. And just remind everyone that this is being recorded and will be available in full on the PCSIG website and the evidence-based website for the indefinite future. So let's go through some of these questions, Mitch. I think the first one is a statement from Jeff. It's just stating that a significant difference in these samples is that Tasso collects, he says, arterial blood. I think that's capillary blood, whereas traditional sampling collects venous blood. And he states these are different at early time points. Just do you have anything to add to Jeff's statement there?
Yeah, just in agreement. I mean, there's papers out there where the data overlaps very well between the two compartments, and there's data out there that it deviates quite a bit. So it's really just molecule dependent and things like that. So it was just corroborating that statement.
So just a question that just came in: will this slide deck be distributed after the presentation? It will be available through the PCSIG website and the evidence-based website. Where if you just type in PCSIG on Google, you'll be able to find our website, and there's a page there with all of the webinars that have been presented and all the webinars that are coming up. So couple of questions from Matthew. Have you considered ISTA 7D shipping excursion stability experiments? Transit temps are not static. So I'm not quite sure what those regs are. But
I'm not either. And you know, when these are shipped, kind of traditionally, it's not going to be a bundle of samples like you're going to see with plasma. It's not going to be 200 samples with, you know, sometimes they might have a device to track the temperature. This is going to be in an envelope shipped in the mail is kind of how these devices are prepackaged. So I'm not sure how feasible it would be to compile all of that data of individual samples. I think we are understanding that it's not static. We're trying to test the extremes. We're trying to test if something isn't under desiccation for a long period of time, if something is, 40 degrees C is quite warm. So we're really just knowing that that's probably not always going to be feasible, and we're testing the extremes to bracket everything in between.
Yeah, and I guess it has, by default, had to go from ambient room temperature to whatever conditions and back again in order for you to analyze it in your lab. So there is some excursion, it's just not controlled. The second question from Matthew was, have you explored within and between device variability with the same donor?
Yeah, so that's reflected in the ISR, Matthew. So all of that would have been done not across multiple devices, but at least from the same donor within the same device. That would be a good experiment to use multiple devices to look at, I guess, inter-device variability amongst patients.
Yeah, absolutely. Question from Aziz. What is the percentage of invalid samples due to imperfect collection?
That's a good question. I mean, it's going to depend on the study indication and how difficult it is to collect that sample. I would say probably about 10%.
Okay, which is quite high. Was that no sample being able to be analyzed at all, or just one of the replicates not being?
Yeah, so I would say one of the replicates being affected in some capacity. So we may have still been able to, and I would say it's more common that the samples are underfilled than overfilled. And so there would still be replicates available. We just maybe not, what's nice about that device in particular is we have an original analysis aliquot. We have a backup in case a batch fails. We have one for dilution and we have one for ISR. So there's really a lot of shots on goal there. So we may not have been able to do ISR on that sample, for example. But I would say you're still able, even if it's affected, in more cases than not, I would say maybe 2 to 3% of samples we received is just like, we're not going to analyze this because there's either nothing here or the device is just soaked. So we can't, those kind of things.
And how would you, just me throwing in my own question here,
Yeah.
Mitch, how would you say that compares with traditional plasma sample, where you might get samples that are hemolyzed or whatever other reason means you can't analyze them? What kind of, is it a higher percentage of failed samples?
Yeah, I would say it's definitely a higher percentage. And I think as the technology grows and is more widespread, that will continue to go down. And, there will be a next version of these devices that maybe circumvents that. Maybe there might be some type of alarm to the patient that knows they didn't completely fill the device, or that it will stop it and prevent the overfill, or things like, some other controls that will prevent that. But I mean, traditional plasma, you're probably less than 1%, something like that, less than 2%. It's not, although it does happen, samples are hemolyzed, and, there might not be any volume in a sample. It does happen. It's not very common.
Yeah. Question from Timothy. Have you looked at plasma protein binding studies using blood collected using the various microsampling devices?
We have not. But I don't see any reason why you couldn't, specifically if you collected using one of the plasma microsampling devices. I don't think there's any reason why you couldn't do that.
Okay, thank you. Question from Dries. Thank you for the great presentation. Can you elaborate a bit more on the addition of stabilizers? It looks very interesting for technology developers to look into this. Does each application stroke molecule require a different stabilizer, or are there a few specific stabilizers that can be selected to cover most of the applications, or is this difficult?
Yeah, absolutely. So I would say, once again, maybe 5 to 10% of molecules require a stabilizer during collection. There are plenty of drugs that are completely stable with just, a K2 EDTA anticoagulant. Sometimes you might want to add an antioxidant or an inhibitor of some type. Esters are very unstable. So, for example, any type of ester drug, I do not feel would be very suitable for this collection, because you're going to need to reduce the pH, add some type of antioxidant, things like that. That, although the drying might help it, I don't think it would completely inhibit it to the point where it could meet validation criteria. So being able to take some type of stabilizer that might be coated in a tube, like a sodium citrate or something like that, and instead of being coated on the tube, coated on the actual cartridge.
Yeah, and I believe some of the vendors are now looking at this, of building stabilizers into their collection devices, and I know that one and maybe more of the vendors have incorporated technology to stabilize nucleic acids, oligonucleotides, etc. So yeah, this is starting to happen. Question from Aziz. Do we need to test hematocrit effect while the volume of blood spotted is fixed and accurate? So I think what it's saying is, now we're collecting a fixed volume of blood, why are we still testing the hematocrit issue?
So I think the hematocrit issue is more so on the analytical side than the loading side. So if you have the 25% hematocrit almost looks pink, whereas the 65% is a very, very dark red, almost a brown. And so when you extract those samples, you might pull some of those constituents off that then cause a matrix effect during your analysis. One thing I didn't touch on was this extraction is much it's more difficult to track than a typical protein crash or something like that, as you're pulling out. So we always say, you need a stable isotope labeled internal standard for your molecules for even just the protein work. This becomes even more important because now you're going to have maybe a little bit more variability from sample to sample with hematocrit. So hopefully the implementation of the right internal standard will track some of those, but the hematocrit effect test is more about on the analytical extraction side of things as opposed to the loading side.
Yeah, absolutely. And yeah, hematocrit, it's known for some molecules can affect recovery and it can affect suppression effects as well. So it's something that definitely has to be looked at for each molecule, even though you've collected a fixed volume of blood. So the volumetric hematocrit effect has gone away. There are still other effects, and drying can affect that as well. Adam's got a question here as well. What is the effect of an overfilled M20 device? Volumetric spots should still fill correctly.
Yeah, that's a good point. So if I were to take that device that I showed, there can be that blood tends to get, like for the Tasso example, that blood you could kind of see was on the outside of the device as well. And if that dries with it, it can form somewhat of an additional crust on the outside of the cartridge. So the overfill could cause a little bit of an extra sample, not from the filling part, but just the additional, like the residual blood that's around the cartridge when it dries.
Thank you. Next question, again from Adam. Do you perform a feasibility method development prior to start of an official method validation?
Absolutely.
Any comments on difficult drug types for VAMS analysis?
Yeah, so we definitely kind of what I laid out that plan. The biggest thing that we're looking at is extraction, recovery, and reproducibility. We'll tend to do the most extreme situation. We'll have something in hematocrit, we'll have it heated, those type of things, and we're making sure that we're still getting reproducible and the same recovery as just a normal sample that may not have been stressed as much. So I would say there is a lot of feasibility work, a lot of making sure that this is going to be reproducible, because we're going to see a lot more uncontrolled conditions than we would with a plasma sample.
Thank you. Another question from Adam. Is 24-hour drying of VAMS test realistic for actual clinical collection conditions?
Yeah, I think the drying component includes the shipment part, because the device will have that label pulled off and then be placed under desiccation, and then will be drying over the. It's not like it doesn't need to be set out at the patient's house for 24 hours before shipment. They're going to collect, remove the label, and then that is going to dry for at least 24 hours. Most of the time, it might be for a month before they receive the samples and things like that. So I think it's realistic.
And Timothy's got a comment on non-specific binding. It says non-specific binding to the device surfaces would be done during feasibility method development. I'm not sure whether that's a statement or a question. But...
Yeah. That's a good point. I think we would, inadvertently see a lot of non-specific binding, just if we saw inconsistent recovery or inconsistent precision and accuracy. That's a good point. It would be good to see that, or to test that specifically. But I'm thinking that it would, if there was a small amount, it would rear its head during a lot of the testing that we're doing.
And it's interesting, isn't it? How far do we go?
Yeah.
Because, with a typical cannula and blood sampling tube and plasma transfer tube, we don't look at that unless we start to see a problem during our method.
Yeah, exactly. And so just his next point about peptides, that was one. We've done some feasibility work for just using this approach with peptides. It was, one peptide, it didn't work very well. So I think we're going to have to come up with a different strategy and have some of those additional considerations for, those mid-sized to larger molecules that have those other considerations like non-specific binding and instability issues.
And I think that brings into another question that we've had offline as well. The amenability of these microsampling approaches to bioanalysis of biomarkers or large molecule therapeutics. Just your thoughts around that.
Yeah, that's a good question. So the presentation was, around a small molecule, but there are people in the literature that are using microsampling for large molecule analysis. There's a lot of clinical diagnostic tests that's done with microsampling that are large molecule. And one thing on our agenda is, we're involved in the large molecule LC-MS space. We use the bottom-up approach to digest proteins and look at signature peptides. So one thing that's on our list is to look at kind of what we're going to refer to as on-tip digestion, whereas normally you might do like an on-bead digestion for a hybrid assay, is looking at putting a large molecule, a monoclonal antibody on a tip and seeing, can we use trypsin to digest this as part of kind of the recovery process and how well that will work. As far as biomarkers go, that's pretty tricky because if it's endogenous, especially if it's endogenous, if it's not endogenous or you can get a stripped matrix that you can then spike in, or, you're looking for a big increase in the biomarker. So the endogenous levels are below your LLOQ, something like that. But the matrix components of this are very critical in the extraction recovery and the matrix effect of your assay. So if you're trying to develop an assay and look at recovery, but you don't have a clean, background in an accurate matrix that you're going to be using, it's going to be difficult to develop that. You could use the surrogate analyte approach where you might spike in a labeled version of that compound and do all of your recovery assessments based on that. But then again, as well, kind of if it is the levels of that are like in the middle range of your assay, the surrogate, you're going to have issues maybe with surrogate matrix for lower calibrators, and just you might have success, but those are just additional things to think about for biomarkers as well.
Thank you, Mitch. And I think we've got time for one more question before we wrap up here. How do you handle samples that need to be diluted?
Yeah, that's a great question. So unlike plasma or wet blood samples where you have ample sample and then you have the control matrix that you can pre-dilute the sample into the analytical range and run, we don't have that ability here. So we utilize, and we include these in our validation experiments, a post-extract dilution approach. So we would extract, in addition, a dilution quality control sample that's above the ULOQ of our method. And we would include what we call control zero samples that have the control matrix, no analyte, but have the same internal standard. After the samples are extracted, then we take a portion of the sample and we dilute it with that extracted control zero sample. That keeps all the matrix constituents the same and dilutes the analyte response into the range. And we show that is linear by doing that with the DQC sample, as well as any patient samples or animal samples that might be above our limit of quantitation.
Perfect. Thank you, Mitch. So I think that wraps up all the questions. So I've just got a couple of slides to go through with people to say thank you, if you just don't mind hanging on for a few minutes. Just would like to advertise the fact that the patient-centric sampling interest group has a number of webinars coming up this year. We've got 11 webinars planned for 2024. I've got the date wrong on the second one there, but these are the ones coming up for February and March. So we're trying to cover as many different application areas and as many different users of these technologies as possible to broadly educate the community on what is available. For those of you that don't know about the patient-centric sampling interest group, it's the passionate group of individuals that are looking to move forward the agenda for the use of these technologies more widely for clinical trials, therapeutic drug monitoring, regular health, wellness, drug testing in sport, wherever blood samples are taken and where these technologies may be a benefit for patients. And we've got a number of activities ongoing. We have an active group on LinkedIn. We have a website. If you want to just hold your phone up to those, you'll be able to get the link straight through to our LinkedIn group and our website. As I say, these slides will be available for free after the event through the patient-centric sampling interest group website and through the evidence base. And I'd finally just like to thank our partners again who enable us to do all of this. And obviously, I'd like to thank you, Mitch, for a fantastic presentation, very engaging. Got a lot of good engagement, a lot of good questions and discussion there. And thank you for all our attendees for putting forward those exciting questions. And
yeah, and if there's any outstanding questions, I'm happy to answer via email. And thank you again, Neil, for hosting me and allowing me to give the presentation.
Oh, it's an absolute pleasure. And thank you all.
All right. Bye, everyone.